insect that lives on these species is a gall-forming wasp. It's a cynipid. The name is Belanite. It has, so gall formers kind of had this really neat interaction with their host plant. They lay eggs. It's a genome-to-genome interaction between the plant and the insect. They induce these three-dimensional tumor-like structures. The organism feeds on nutrient-rich tissue within these tumor-like structures, and then they emerge. And so they have this cyclically parthenogenic life cycle where they alternate between a sexually reproducing generation, seen here in the upper right-hand corner, and so asexuals will emerge from here, and then there's a sexually reproducing generation on roots that are underneath the ground. Individuals will emerge from here in the spring. So we're going to concentrate on this generation to calculate reproductive isolation because this is the generation that sexually reproduces. And so when they emerge, they lay their eggs into newly flushed leaves. And so they can only induce galls in leaves that are really, really young. So as soon as the bud breaks, these live oaks are deciduous, so they drop their leaves. But as soon as the bud breaks, they can begin laying into new leaves. And this resource is only available for maybe about 48 to 72 hours. So if they lay into it after that time, they cannot induce a gall. So it's a very, very ephemeral resource. And interestingly, coinciding with leaf flush is also the production from the same buds of catkins or oak flowers. So that's going to become important in a second. So we've also documented through a number of studies host-associated differences in reproductive isolation of bee treaty, a number of them ecological, morphological, and behavioral as well. So QG produces larger leaf galls and root galls than QV. There's strong habitat isolation. Individuals show a preference for their natal host plant. They don't seem to like as much their non-natal host plant. There's mating preference where individuals prefer to mate with individuals from their natal host plant. And so collectively, this provides strong evidence that divergent host plant use may promote ecological divergence. So the hypothesis that we explicitly wanted to test was that differences in host plant phenology, the timing of bud break, result in parallel differences in the phenology of emergence of sexual generation bee treaty. And so we kind of took it trophic level by trophic level. We wanted to document this in the plant, then we wanted to document this in the insect, and then we used this information to actually then calculate reproductive isolation in this system for the insects. And so the first thing we did is we summarized herbarium records from 3 herbaria across an 86-year non-continuous period from photographs. And so from these photographs, we can tell the species of the plant, we have the location, the collection date, and then we have the presence or absence of flowers. And so remember I told you that as leaves unfurl, buds break, flowers come out too. So this is a proxy for leaf flush. So in total we had 110 QG plants, 128 QV plants. The number here, 24 and 28, is the number that were flowering, the numbers we could score for flowering. This was across years, and most notably, I'm not going to show this data, but the surveys were done throughout the year, number one. And number two, we note that flowers were only observed during the typical time of oak flowering. And so here are the results for that. There's about a 38-day difference between flowering time. QV flowers earlier than QG. This is significant. We found a moderately significant latitude effect here. So next we wanted to go out and monitor leaf flush or bud break at a number of individual sites, 7 sympatric or parapatric sites. These are high gall density sites. This will become important in a second. So over about a 2-month period between March and April, we went out 14 different times every 2 to 4 days. We would go up for every tree at every site. We would randomly choose 15 branches. We counted the number of broken and unbroken axillary buds on about 120 centimeter length of branch. This culminated in monitoring 41 trees, 75,000 leaf buds. And this is what this looks like. So the x-axis here is cumulative frequency of bud break. And then we have date here. These 4 sites are sympatric sites. This is all the data pooled across sympatric and parapatric sites with the means in these darker lines. The gray is QV, the red is QG, and you can see that QV is flushing leaves earlier than QG. It's significant, and we don't see a difference between sites, so this seems like it's maintained across different sympatric sites. So we used these same sites, and we wanted to monitor emergence of these organisms coming out of galls. We did this in 2 ways, at least a subset of these sites. We went and every time we monitored leaf flush, we put out yellow sticky traps. These insects are highly attracted to yellow sticky traps, so every 2 to 4 days, we would collect old traps, put new ones out. And then we also made bulk collections of root galls and brought those back to the laboratory. So we have estimates of bee treaty emergence not only in the field but in the lab. And remember, these things don't live very long. Their average lifespan is about 2 or 3 days. So if we were able to find differences, it's probably a strong temporal isolating barrier here. And so here's what this data looks like. The top is for the field-based stuff. The bottom is for the lab. Again, date and the cumulative frequency emergence. I've got it broken up by site, but the gray scale colors are for QV. The red scale colors are for QG. We can see the QV organisms in both the lab and in the field are emerging earlier than QG. Right here I've summarized the difference between 25, 50, and 95% emergence for both the lab and the field, and it's about a 2-week difference. And so this is just a correlation here, the mean date of bee treaty emergence and the mean date of bud break. We can, and I've just fit these lines here so you get an idea of what the shape is, and it's both for the laboratory and the field component here, but we can see these are highly correlated, just providing strong evidence that temporal isolation of the plants cascades across trophic levels and produces temporal isolation in the insects that use these plants. But we can take it a step further. We can use this fancy equation here and actually calculate values of reproductive isolation. You don't need to get enamored with the values in the table here. Just note that reproductive isolation between any pairwise QV and QG sites can be quite high. Temporal isolation above 90%, and the averages here are 55% and 82% when you use the laboratory and the estimates in the nature. And so we can actually use these estimates of temporal isolation, and we can combine them with our previously published estimates of both habitat isolation and sexual isolation or mating isolation to calculate total prezygotic reproductive isolation in this system. So what this table shows you is estimates of the lowest, average, and high values, and the numbers in the parentheses here are just the percent contribution. So both in the average and the high, we see that temporal isolation differentially contributes to total reproductive isolation, and that total reproductive isolation in this system can be quite high. So I had a colleague earlier today, Amanda Driscoll, who gave a talk on the population genetics of this system, and they're verging on different species level statuses, those on QV and QG. So this could be a mechanism of reducing gene flow in this case. Going the wrong way. So I'm going to skip the summary here. So these two sister species of oaks are home to many other different gallformers, all using ephemeral tissue, leaves or stems, which is another ephemeral tissue. And they're attacked by this diversity of natural enemies. So the kind of question is, could we see that cascading temporal reproductive isolation is contributing to multiple species? And so this is Lin Yu Zhang, a PhD student in Scott Egan's lab at Rice. And so what she's done is kind of calculated this for an additional 5 species of gallformers plus 3 natural enemies that attack these gallformers. In all cases, we see that QV, or those insects developing on QV emerge earlier than those insects developing on QG. So this could really be an important biodiversity generator in particular systems where you have long-lived hosts, short-lived insects that live on them that use ephemeral resources. Sorry I ran a little long. I'll take questions. I'll be out in the hallway.